All right, so we know PFK is the rate controlling enzyme for glycolysis, right? We have a video out there that shows us how, inhibitors and so forth can change the activity of the enzyme. But now we're going to actually look at the structural details of how PFK is regulated. Okay, we know PFK regulates glycolysis, but again, this is the how it's able to do so. And as we've learned before, two letters that are important for us understanding what's going on with enzymes or proteins are R and T. So R state and T state, right? So R state represents a conformation that is conducive to binding substrate. T state is a conformation that is not conducive to binding substrate. So I'm going to get to what we have over here on the left in a minute, but let's just write that down and remember that basically we're going to have a T to R state transition, conformational change. We're going to look at the colors here for a minute, but these colors tell us that the T state of this protein are what's shown in blue, and the R state is what's shown in red, and reminding us that R state, right, is a conformation that's conducive to binding substrate. So conformation, conducive to binding substrate. Okay, so let's look at this conformation that we have here. So normally we've been looking at proteins, right? And we see sort of these bubblegum structures, these space filling models. What we have here is not a space filling model. It's just a ball and stick model. And it's not even the whole protein. It's just a portion of the protein that encompasses two important regions. And I'm going to highlight what those are here. Over here, what I'm highlighting in green, this is going to be the enzyme active site. Okay, so this is the enzyme active site. So this is, again, where substrate binds and where chemistry happens. And I'm going to have another site that I'm going to circle over here. Okay, this is what we call the allosteric effector site. Okay, we're going to talk about each of these in turn. But one other thing I want to highlight is we see some of these arrows that we have here. These arrows that just kind of show things moving. Okay, these arrows, okay, I'm just going to kind of label them here and then label them down here. Okay, these arrows are going to represent what happens during a process that we're going to call helical unwinding. So remember, when we have conformational changes, that's changes in the shape. So we have alpha helices, we've got beta strands that interact to make beta sheets, all of that we learned about with protein structure. So helical unwinding is taking that telephone cord, if you will. Remember, when we have an alpha helix has the side chains that point out and down around the outside of our alpha helix. So if I take and I unwind that helix, maybe you can see this in your head, we're really changing the three-dimensional space where those specific amino acids are. And so what we see here is there's some places in this structure who going between T state and R state that happens with helical unwinding really overlap, right? T state, R state, the same, okay? A lot of these kind of show not much change that we have here. There's some other places that we have a little bit more movement, a little bit more change. But here's the money part. Here's the part that's really important. This is where we have a significant amount of change in our structure here. And what's going to happen here with this helix that unwinds, we are really swapping out. Look over here in the enzyme active site, in that red T state structure, we have an arginine residue. But when we do this change in conformation, so again, from T state to R state, going from T to R, I should put this over here. When we go from T to R, this is when we have this helical unwinding. So helical unwinding is what causes that T to R transition. I don't need you to remember whether it's winding or unwinding, but we have a conformational change that goes from T to R. And what happens specifically at the enzyme active site is we're going to have let's talk about first the T state has a glutamic acid 161, And so it's the adjacent residue, 162, as an arginine that now gets to slide into the enzyme active site. So by unwinding this helix, we're changing which residue, two adjacent residues, which one gets to sit in the enzyme active site. Okay, so one other piece to highlight here before we sort of put this together. One other piece, one other thing that we see here. Let's go ahead and we'll highlight this in red here. Okay, this is our substrate. This is our F6P substrate. Okay, so we've got our substrate that wants to, or not, sit in the enzyme active site, right? R state means we've got an active site that's conducive to bind that F6P. T state means we have an active site that's not. So we're going to put all of this together here with a little chart, okay? So if we think about what amino acid is present in the enzyme active site when we have T state, remember T state being blue, look over here, we've got a glutamic acid residue. So glutamic acid is in the enzyme active site when we are in T state. When we unwind that helix and we change to R state, now all of a sudden we're going to have that arginine that's in the enzyme active site. Okay? And let's think a little bit about what functional groups we're going to have in those different amino acids. For glutamic acid, it's going to be a carboxylic acid. And importantly, that means it's negatively charged. Okay? When we put arginine in there, and I'm okay if you don't exactly remember the structure of arginine, okay? It's a guanidino group. But importantly, it is a positively charged protonated amine, okay? So we're going to have essentially a swapping out of a negative and a positive charge. So let's ask the question, how will that affect our F6P binding? Okay, so let's look over here. Remember the structure of F6P. Fructose is not a molecule that's charged. Fructose is uncharged. It's polar, but it's not charged. But when we put that phosphate on that 6th position, all of a sudden it accumulates a negative charge. Okay, so we've got a negative charge here, and let's think about whether it wants to sit next to a positively charged arginine or a negatively charged glutamic acid. So again, F6P itself is negatively charged. So when we think about what's happening and whether or not we're being conducive to binding our substrate, when we've got glutamic acid there, nope, significantly decrease our propensity to bind our substrate. When we've got that positively charged guanidino group, yep, much more interested in binding our substrate. So the reason for this is all electrostatic. It has to do with whether there's electrostatic repulsion, because we've got two negative charges that would be in the same locale. That's not desirable. Or we've got attraction, where we're going to have a positive and a negative that are going to, again, make that enzyme active site conducive to binding its substrate. So again, let's look at this over here. Remember, red is R state here. So when we're in R state, we've got this positively charged arginine group that's going to make it easier for that F6P to bind in the substrate, in the enzyme active site. When we are in T state, there's a glutamic acid residue that's there. That's going to form a repulsive interaction with the substrate, again, making it not conducive to bind its substrate when we're in T state. So some structural details there that let us to understand how a conformational change is going to control whether or not we are conducive to binding substrate or not. The last little piece here I want to map on is what happens with this allosteric effector site. Okay, two things that we can sort of see here is we can see either ADP or ATP binding in this enzyme active site, or I'm sorry, this allosteric effector site. Now, one of the things that can happen is ATP, let me move these down just a little bit, give myself a little room here. ATP is going to allow us to lock in the T state. Now think about this, we know this. We know that ATP is a potent inhibitor of glycolysis. So by locking PFK into the T state, that means we are inhibiting glycolysis. ADP will lock it into the R state, okay? And I'm not going to get into the weeds of the structural details here, But if we think about what's the difference between ADP and ATP, it's a single phosphate. But that is enough to sort of control whether or not we have a molecule that locks in the T state, okay, and allows for that glutamic acid to stay in the enzyme active site. And so F6P is not going to bind productively. Or we have helical unwinding that switches in an arginine residue, all of a sudden makes for a favorable electrostatic interaction between a glutamic acid residue and that F6P substrate. And all of a sudden now we're ready to do chemistry. All right, so some structural details about how PFK is regulated to control flux through glycolysis.